Today I'm going to talk about some research I did as a part of my dissertation at UT Austin in the lab of Harold Zacon. So part of my dissertation, I was really interested in the process by which a redundant duplicate gene evolves a novel adaptive function, a process called neofunctionalization. And in particular, I was interested in how the neofunctionalization of duplicate genes contributes to the evolution of novel phenotypes in organisms. So my system of study was a group of weakly electric fish called the gymnotiforms, which are found in fresh waters of South America. These fish generate pulses and waves of electrical signals which they broadcast into the water to communicate with conspecifics, as well as to navigate in a nocturnal environment. And the organs that they use to generate these electrical pulses, called the electric organ, is comprised of electrocytes, which are highly derived cells developmentally and evolutionarily from muscle cells, or myocytes. They're multinucleated, and early in development they have sarcomeres, which are then degraded by the time the electric organ is finished developing. Along with having being highly derived morphologically, they are also highly derived electrophysiologically, where they've co-opted the molecular machinery that generates action potentials in skeletal muscle for the job of generating the electrical signals that they broadcast into the water. And interestingly, the waveform of these electrical signals has greatly diversified in this clade of species, driven by selective pressures such as predation from electrosensitive predators, as well as sexual selection on the shape of the waveform itself. And so this diversity of electrical signals implies that we can determine interesting evolutionary patterns in the molecular machinery behind the electrical properties of excitable cells, namely the voltage-gated ion channel family. And there's one type of voltage-gated ion channel called voltage-gated sodium channel that has played an important role in the evolution of the electric organs. And in particular, the muscle type of sodium channel, the type 4 sodium channel. So in vertebrates, there is one muscle-specific sodium channel, but due to an ancient genome duplication in teleost fish, they have two copies of the type 4 sodium channel, which are broadly expressed throughout the clade of teleost. Now in the weakly electric gymnotiforms, the type 4AA sodium channel is actually lost from the skeletal muscle and has been compartmentalized in the novel electric organ, where its sequence and electrophysiology indicates that it has gone through neofunctionalization for specific function in this novel organ. But there is an exception to this pattern within the gymnotiforms, and that's in a clade of highly successful electric fish called the apternodids. In their muscle, unlike the rest of gymnotiforms, they still express both paralogs, and they actually express quite a bit of the type 4AA paralog. And this could have something to do with an even more novel electric organ that has evolved in this clade. So like the rest of gymnotiforms, they, early in development, have a myogenic electric organ that is innervated by spinal neurons that govern its rate of firing electrical signals. But by the time they reach adulthood, they've lost the myogenic electric organ and replace it with a morphologically highly derived spinal neuron that generates their electrical signal. investigating the role that the voltage-gated sodium channel gene family played in the evolution of this new neurogenic electric organ, given the interesting role that this family played in the evolution of the myogenic electric organ. And so in teleost fish, there are 8 different voltage-gated sodium channels. And I used RNA-seq to assay the patterns of expression of these sodium channels. So I assembled and analyzed the transcriptomes of the muscle and the spinal cord, where the neurogenic electric organ is located, in 3 different actinoids with the neurogenic electric organ. One genotiform that has a myogenic electric organ in adults, and then one non-electric cousin channel catfish. And so in muscle, we see that patterns consistent with previous research, where both paralogs are expressed in the skeletal muscle, and those are the only sodium channels expressed there. And then consistent with previous research, we see in the myogenic electric fish, only the type 4AB paralog is the only sodium channel expressed in their skeletal muscle. But in the neurogenic actinoids, in all 3, we actually detected 3 different type 4 sodium channels, and one of them being a new duplicate that's very similar to the type 4AB paralog. And looking in spinal cord, we found the patterns of expression are consistent with expectation in previous research, where expression is dominated by type 8, type 2 sodium channels. And we see that catfish and the myogenic electric fish spinal cords are very similar. But in the actinoids, again, we see that this new paralog, what I'm calling the type 4AC sodium channel, is also expressed in that organ as well, which is a very surprising result to see a sodium channel that is highly conserved as a muscle sodium channel among vertebrates, to see it expressed in the spinal cords of these species. So this gave us the natural hypothesis that perhaps this new duplicate sodium channel gained expression in the neurogenic electric organs of actinoids and has evolved for function for that novel organ. And so to explore that further, I used a random effects branch site model of codon evolution to fit a DNDS ratio to each branch in a gene tree. And so in constructing this gene tree, using the type B paralog and the new type C paralog as well, in genotiforms as well as non-genotiform species, was able to time the duplication or the duplication event, which I'd indicated with an arrow on that phylogeny. And then just looking at the evolutionary history of the selection pattern of the DNDS ratio, going from the most recent common ancestor of the actinoids and the zebrafish, a non-electric species. We see that throughout the evolutionary history of both the 4AC and the 4AB duplicates, their shared evolutionary history shows a very conserved pattern of selection with the DNDS less than 1. But at the duplication event indicated by the arrow in these 2 plots, we see the type 4AC paralog, which is expressed in the spinal cords of actinoids. We see a sudden acceleration in its evolutionary rate, followed by F4AC paralog happened after the evolution of neurogenic electric organs. And so we predict that some basal plates of the neurogenic apternoids do not have this duplicate gene. And there are some interesting differences in the electrophysiological properties of these different fish that could be explained that I didn't think I had time to get into, so I didn't. And then finally, SCM4AC likely participated in the electric signal adaptation in apternoid species. So with that, I'd just like to thank Yin Liu, the technician in the Zaycom lab, who was a lot of help on this project, and then my graduate advisor, Harold Zaycom, and thank you for listening. No, I didn't. I didn't have the genome for these species, but there was recently the genome for the electric eel was done. That I could potentially look into that question. I think that'd be interesting.
So how comfortable are you guys that this 4AC isn't splice variant of the 4? I mean, I know that the type 4 doesn't typically have splice variants, but just because you just have the RNA-seq.
Yeah, it's a good question. So I looked at, I aligned this new duplicate with the type B, and I see polymorphisms are evenly spread throughout the entire gene. So I'm pretty confident that it is, in fact, a different gene. Yeah.
Were there any, since you did RNA-seq, were there any other genes that showed a similar expression pattern that might be additional genes?
There was nothing that showed this. So there was nothing that showed such a stark pattern. So I kind of got distracted by that. So I haven't looked in too much depth at other ones, but it doesn't. Everything else seems pretty similar, actually. So that's a good question. Okay, thank you.